Hi, everybody. This is John Valdez. I come to you from InsightRX. I am actually the Senior Director and Product of Customer Experience at InsightRX, and I'm happy to have you all here for this learning lounge on Beyond the Vancomycin Guidelines, how precision dosing intelligence creates a cycle of continuous learning and improved outcomes. A quick one-liner about me. I'm an ID pharmacist by trade. I actually spent about 10 years as an ID pharmacist in the Bay Area for both adult and pediatrics before I came across InsightRX and found this type of thing that they're doing that is just fascinating and exciting. So I wanted to share sort of a story about this cycle of continuous learning and how it feeds into things to get you an idea of what this future might hold on here. So we have Andrew Su, who's with Marketing InsightRX, also on the line. He's going to be managing all the questions in the chat. If you didn't get a chance to read that, if you do have a question, please put it in the question and answer section. We'll do our best to address those at the very end. We should have some time. And let's get going on it. So what we're going to be talking about here is precision dosing intelligence. And we'll talk about what that means. But the core idea is that the data that we have shouldn't be static. We should be able to continually advance and improve our data as we go along. So as we talk about that here, we're going to be talking about three pillars that get you to precision dosing intelligence. The first on this left side is dosing decision support. And we're going to be using InsightRX Nova as an example for dosing decision support. But we're going to start with this unusual patient case. And then we're going to talk about some things that help to lead into optimizing that decision support system to use the best data available to help you make good decisions for your patients. And we'll cover Dose Assist, which is a feature on there to help, but also some more core things like model-informed precision dosing, or MIPD, and map Bayesian forecasting. From there, we'll talk about how that case in this story leads into real-time analytics. And we'll touch on the InsightRX Apollo platform here, although I'll remind everyone this is not intended to be a demo. We're just going to use these things to explain what we want. If you do want to see a demo, we have a booth open right now. Feel free to stop by or contact us for that. But in the story, you'll see how in this unusual case, those findings were then applied to a greater audience within the institution and led to an exploratory drill down to be able to find out what's going on and whether there's something real. And in this case, they did find a signal, a discovery of a signal. And with that signal, it led to the third pillar of precision dosing intelligence, and that's going to be continuous learning. And that's the process of using this signal to then do a validation of data on a larger scale, and in this case, actually help to improve the underlying model that exists. It's a really fun story. Obviously, I'm super excited about it. I hope you all enjoy it as we go. But before we get going, let's just make sure we all talk about the same thing with precision dosing intelligence. And I'll start by talking just about precision medicine as a whole, because that term has become more popular recently and can mean a lot of different things. So just to define it here, when I say precision medicine, I mean using individualized patient factors like physiology, behavior, and genetics and genomics to help optimize patient care. And that could be helping to define risk of disease. It could be helping to choose therapy, or it could be helping to define what dose should be used. This is getting a lot of press right now, but it's not particularly new. Like if you think of like an HIV, right? HLA-B5701 that we test for abacavir use. That is using a genetic component of a patient to determine whether or not a certain therapy can be used. And that's precision medicine. What we're going to be talking about today is precision dosing, and that's just a subset, right? It's using physiology, behavior, genetics, genomics to help define the optimal dose for a patient. And again, a lot is being said about this now, but this is not a particularly new idea. There is just a one-liner saying precision dosing is the individualized dosing for a unique pharmacological profile. But again, this is not particularly new. We do this now to a certain extent. It's just we've started and for some reason stopped along the way. So if you think about in practice now, we don't give everybody the same size dose, obviously, right? You all know this. We dose based on body weight. If you think of vancomycin, if you follow the package insert, which very few of us do nowadays for most indications, but the package insert would say 4 to 6 mg per kg of total body weight. And so different size patients get different size doses. Not only that, but we dose based on body habitus, right? And body habitus is saying that two patients with the same weight may not require the exact same dose. If you've ever used adjusted body weight, which actually a lot of us are turning to now for daptomycin, That's an example where we're saying it's not just the weight of the patient, but the tissue composition that should determine how we do dosing. It's individualized dosing for patients. And then perhaps most commonly, organ function, right? I mean, for pharmacists, renal dosing, this is bread and butter. This is what we do. And that's a form of precision dosing. We're saying depending on the functionality of someone's kidney, their ability to excrete the drug can change, and we have to individualize dosing for the patient based on that. The issue is that we have these underlying things that have existed for some time, but we've sort of stopped along the way and accepted that we've gone far enough and accepted the fact that there are going to be flaws and just started to work around them rather than continuing to try and improve. And I'll give you an example of that. So the example I have is creatinine clearance, which I'm sure I don't have to explain to people what creatinine clearance is about, but just to make sure we're saying it the same way, obviously creatinine clearance is a surrogate marker for GFR, glomerular filtration rate, and by using a product of muscle breakdown, which is creatinine, which flows into the plasma and then flows into the kidney. We measure the plasma concentration, do a little bit of math for a calculation, and get an estimated creatinine clearance. But the issue is with that, while it's pretty close most times, we know that there are some issues with our standard measure, right? I think most people know that creatinine is, for example, actively secreted into the proximal tubule, which means that creatinine clearance for most patients is a little bit higher than true GFR. But we've adjusted for that. Manufacturers and drug makers have adjusted, and sometimes they say to dose based on creatinine clearance, right, instead of true GFR. But there are other things that are a little tougher, right? I mean, we know that creatinine production is a product of muscle breakdown, and that's going to mean that if your muscle breakdown is outside of what is expected, it's going to impact our creatinine clearance. A great example is a bodybuilder who is creating and destroying large amounts of muscle every day. That person is elevating their creatinine production, and it may be falsely depressing the estimate of creatinine clearance we have because of the high creatinine levels. On the other side of it, people with low muscle mass, I mean, how many of you out there round up serum creatinine in the elderly patients, right? Based on the idea of low muscle mass, low creatinine production, those type of things. It's something that most of us are trained to do, although we may be trained slightly different on what the rounding should be, but it's a fudge factor that we put in there. And it's just been accepted that, well, there are limits to what we do and we're going to work around them. And the whole idea behind precision dosing intelligence is we shouldn't have to just accept this static data and say we're going to work around it continually. We should be able to improve the data continually with this cycle. And that's what we're going to talk about. So let's go right to that first pillar, which is dosing decision support. And again, we'll be using InsightRX Nova. I'm not going to do a real demo of this. I'm going to do a couple of screenshots to make sure that we have technically safe presentation. But first, let's get into this unusual patient case. And this was based on someone that was presented to us by one of our partners, one of our customers. So this is Stella Kicken. Stella Kicken is a 67-year-old woman, 5'3", 125 pounds, serum creatinine of 0.6. She is pretty healthy. She has hypercholesterolemia, but she is socially active, physically active. She goes to Zumba 4 times a week, loves playing with her 2 grandkids. Unfortunately, over time, as might happen, her knees didn't keep up with her active exercise and she had to have a knee replacement. And more unfortunately, with this knee replacement, she got an infection in it, had to have a prosthetic joint infection. The joint was removed. And as you can guess, since we're talking about vancomycin as our core, it's going to be MRSA. We're going to have to use vancomycin. And I think as everyone who is on this Learning Lounge knows right now, the new vancomycin guidelines say to do AUC-based dosing for severe MRSA infections. We're going to categorize this as a severe infection in this case and move forward with a dosing decision support system. And there's a few things that play into this. I'll mention dose assist, which will be one feature we see. But the core idea behind dosing decision support is to use the best available data to help support your decision-making in real time with patients. And so we're going to use InsightRX Nova in this case, and I'll just pull up a screenshot of that. Very quickly to just walk you through the area. On the left-hand side of this is the patient summary section, showing all the patient data, height, weight, age, all things that can be imported from a chart if you have an integrated version. The drug monitoring section, this is going to be bumped down shortly, but this is where you'll see drug doses and serum creatinines and levels. The top right, also, this is going to expand in just a moment. This is the analysis panel. It's sort of the brains behind the system. You get to see what InsightRX is thinking. And then finally, in the center section is dose information with show regimen options. And I don't think you can see my mouse, but I'm going to click on show regimen options right now to get things going for this patient. Great. So we're now seeing the settings on here, and there's dose assist on here, and I'll talk about that means in a moment. But what I want to point out really is the fact that we're using a model down here below, right? You can see under modeling settings, we have a model. In this case, it's the adult model for the general population that was made by Goethe et al. And this is a good model. Among our models that exist, this is probably head-to-head for the top two between Goethe and Thompson are always in a battle. But it's a strong model, and it's probably the best available data that we have, and we're going to use that best available data to help us guide the initial dosing on this patient. We're going to use the model to inform our precision dosing. So I'm going to click OK on here. You're not really seeing me click OK, but that's okay. And then the whole screen populates out. Obviously, you can see now a time concentration curve. You can see some regimens appearing in here. And I'm actually going to zoom in on the regimens right here. And you can see now we have a series of regimens listed, right? And each of these, I'll just sort of walk through from left to right on the top one. So you're seeing that 500 milligrams given every 12 hours, infused over 1 hour, is predicted to give us an AUC at steady state of 420, a steady state trough of 14.3. This PAUC is the probability that the AUC will be at least 400 as a marker of efficacy. In this case, it's 56% with the data that we have. The PCONS is the probability that the trough concentration at steady state will be above 20 as a marker of toxicity, in this case, 16%. And then finally, the final column, TOX, is short for nephrotoxicity, and it's the chance of a nephrotoxic event in patients like this, given this regimen, based on a study by Tom Lodis and his group back in 2009. So you can see we have a series of regimens on here, and this is actually a good chance to talk about dose assist, which is another way to support your decision making. What we heard from people in a lot of the feedback was people said, it's great that the model here is showing me what I can do with different regimens, but what we really want is for you to tell us what regimen to give the patient. Just give us the answer. And the response is, first of all, legally, we can't do that. We can't tell you what to do. But more importantly, we don't want to. This is designed to be dosing decision support. Dosing decision support should provide you with the best available information to help you make the best decisions possible. But the compromise on that is to say, well, what we can do is eliminate all the fluff and all the chaff and just give you data that is most likely to be useful to you. So the way Dose Assist works is our clinical team, clinical pharmacists got together and created a sort of a rank preference list of regimens from 1 gram Q12 at the top all the way to 2 grams Q6 at the bottom, and ranked them. And so Dose Assist goes through these regimens and picks regimens starting from the top down that are the most likely to get you in your target range. In this case, an AUC of 400 to 600 and a trough of 10 to 20. So they may not all be the ones you want to use, but chances are of these 4 or 5 regimens, one of them will be good for you. As we go through in this case, the top one, 500 Q12, an AUC of 420, that's okay, but this probability of an AUC above 400 is only about 50%. I kind of, in this case, want to go a little more aggressive with her. The next one down, 1000 Q24, also has a similar AUC, but as we go down 1250 Q24, that gets me an AUC of 525. My PAUC is up to 81%. My toxicity risk also goes up, but I, as a clinician, know this woman. She's healthy. She doesn't have a very high risk of kidney injury at all. She's not getting contrast. She's not septic. So I think I'm willing to take that risk in the beginning to ensure that we have a good AUC. And as we get levels, we'll be able to refine things as we go. So great. Let's pick 1250 Q24. And this is what they did in this case. And that's selected now. And you can see on the right side now, you see how the time concentration curve over there is kind of going from low to high. This is a great example of how long it can take to get to steady state, and a good case for giving a loading dose. And again, this is dosing decision support saying, hey, just looking at this, you may want to give a loading dose. According to the IDSA guidelines, I could give up to 30 per kilo, but I'll tell you honestly, I'm becoming an old type of dude that 30 per kilo still scares me a little bit. I have to get comfortable with that. So we'll go down a little bit from there and maybe just click on the loading dose square and do 1500 milligrams, which is pretty close to 30 per kilo. And you can see on the right side in that chart with the time concentration curve, the red line, how we got a loading dose in there and it got a little closer to steady state. I'm actually just I'm gonna backtrack one so you can see before and after. Here's before, and then after gets us closer. But the fact is, it's still gonna take a little while to get to where we wanna be on her. So I'm gonna show you another trick that we can do that's gonna, again, it's helping to support our decisions on what to do for this patient. I'm zooming in here on the analysis panel, and if I hover over this time concentration curve, I can actually see the predicted concentrations at any given point in time. And I can see that for this regimen, where my steady state concentration is expected to be 15 for the troughs, I can know that it's going to be 15 at 8 o'clock tonight. So after this load, why don't I just start my maintenance regimen at 8 o'clock tonight? It'll get us to steady state right away. Why not, right? So let's simulate that right now. And you can see on the right side with that time concentration curve, look, a load and then a maintenance dose pretty quickly afterwards, and boom, we're there. We're at steady state. So this is what was done in this case. And you can see this is sort of using a model and predictions from the best data we have available to help guide your decision-making process in your patients in real time with, again, just the best data available. So this is what was started, but of course, this isn't the end of the story. This is just the start. It's not even unusual at this point. Let's get into that fun part. So this Stella Kicken was given her 1500 milligram load. Her first maintenance dose was given, and then they decided to get a level right after that first maintenance dose, because for one thing, we're already at steady state. But second, with these Bayesian platforms, you can really get a level almost any time that you want to. And unfortunately, that level came back at 9. And since we were expecting it to be at steady state, we would have thought it would be a lot closer to 15. So that's a surprise. We're going to have to deal with that. So I'm going to go back to InsightRX right now, and you'll see all the data now is included in here. You can see down at the bottom in the drug monitoring section how we have serum creatinines and doses and levels and all of that. Again, in theory, this could be imported from a chart if you're an integrated system. But I'm going to point your attention to the analysis panel on the right side. And the first thing is the bottom section, actually. I want to show you down there. You see how we have the first dose and the second dose pretty quickly after. And this level came back low, and there was an adjustment to the green line, which is historical data, in order to fit to this level. This is Matt Bayesian forecasting, which I won't go into now, although if you ever want to talk about it, call me up. I'll go on for hours about how cool Matt Bayesian forecasting is. But you can see it made an adjustment to individualize it based on this new data that we have. The other thing I want to point to is up top, where that red box up there. We didn't talk about this before, but these are PK parameters. So on the left, you see clearance, volume of distribution, and half-life. And the next column over population, the population values are basically what the model would have predicted. The Goethe et al. Model says for this woman, Stella Kicken, with her height, weight, age, gender, serum creatinine, etc., we thought her clearance would be 2.1, her volume distribution would be around 48, half-life would be 18 hours. Once we got this level back, things are a little bit different. And in that individualized column, that's the Matt Bayesian fit saying, well, her clearance is higher. And that's the point to emphasize here, because her trough was low. So of course, to make that fit, her clearance is probably higher. How much higher, 3.5 versus 2? Well, on the right side, you're informed through this population bell curve saying that this little dot resting on it, you know, if she's perfectly aligned with what we expect, it'll be top middle, but you can see it's moving down the curve toward the right side on there. And to the point actually where dot is becoming yellow as a caution saying, this patient is now considered unusual. It's not what the model would expect. Now that yellow dot means we're still able to accommodate it with the model. If it was really not able to be accommodated and that unexpected, it would be red, but it raises questions, right, about this patient. Who are they? Why is this happening? The other thing I want to point out on this is because the level came back low and her clearance is higher than expected, we have updated predictions on these regimens. So her previous regimen of 1250 milligrams Q24, well, now it looks like that AUC is going to be 364, below our target of 400, a trough that's 8, which is below what we were looking at, although questions can be raised about whether we care whether the trough is that low. And things just aren't looking as good. In fact, it looks like maybe we want to switch to 750 milligrams Q12, at the least for her, to get up to a higher AUC of, it looks like around 430, between 400 and 450. So we've discovered something new about this patient. She wasn't who we expected, and we're able to adjust. And they did, in this case, they changed to 750Q12. They got a repeat level within 24 hours, actually, just because they wanted to have confirmation. And it ended up matching what they thought. She went on, got a good course, and everything was just fine for her. But again, it raises the question for us of, why is she unusual? And this pharmacist who was working with it, noticed it, talked to other pharmacists in the same hospital and said, are you seeing things like this? And they said, yeah, this model seems like it does really well in general, but in this type of patient, these older, elderly patients who are healthy and have a low serum creatinine, it seems like we undershoot a lot, and it seems like we have to adjust up. And now, historically, what we would do from there is say, there's a deficit. I guess we're going to have to fudge it and just pick something that predicts a higher thing on there. We'll fudge it, or maybe it's not even an issue. Keep going, right? But again, the whole idea behind this in precision dosing intelligence is that's only the beginning of what we do, is saying maybe there's something we want to adjust to. And that brings us to this second pillar once we come back here. The second pillar is real-time analytics. And I'll just talk about what I mean when I say real-time analytics, because it's, again, can mean a lot of things. When we say real-time, we mean analytics that are available on demand, at a moment's notice. I think we're used to thinking of analytics and reporting as requesting a report from maybe our IT department, if we have one and we're lucky, or from a resident doing chart digging, and trying to probably take maybe even a few months to get data across 6 months to a year or 2 years, and then processing that data and cleaning it up, and eventually, again, months or a solid residency project later, getting information about what was happening about a year ago. And if we find that out, we can say, it looks like we've been doing something that could be improved, or we're doing a good job for the past year or two. With real-time things, the idea is you look at it at a moment's notice. You open something and you have analytics available to you at a click. And what that means is you can see the impact of interventions as they unfold. If you're switching from cefepime to Zosyn because there's a shortage, you can see, are we having an AKI impact as this is happening? You can then determine whether you need to take action now, right? Not a review 6 months from now to say, we've been doing it wrong for a while. We can improve right now. The other thing you can do with analytics, sort of in this modern day, is to be able to drill down on that data easily. And that means being able to examine the performance in different patient populations. So again, we can look at patients in this Zosyn example of patients who are co-administered Zosyn versus co-administered cefepime. How are things relating with AKI? We can look at different patient populations of people who are obese if we have a suspect. We have a patient population right here that we want to look at of patients who are elderly with serum creatinine of less than 1. You can also look at performance of different users in different groups. I have multiple sites and I want to see how my different hospitals are doing. I want to see how they're doing on different shifts in the ICU versus on the med-surg floor. If you want, even individual users or pharmacists who may need some education or help, you can look to see how they're doing individually. And all this can be used to see that you're doing a good job in areas or identify areas for improvement, again, in real time. So that's the promise of real-time analytics. I'm going to show you the example that we have here, which is InsightRX Apollo. Again, this is not a comprehensive demo. We do have a booth set up if you want a demo, or you can contact us at any time. This is just going to be a few screenshots that tell our story. So if you remember, in this case, we have this woman and this suspicion that people who are elderly with serum creatinine less than 1 were undershooting. And so the first thing I want to do, obviously there's a lot of information here. I'm going to focus just on this Pharmacokinetics tab, which is about sort of target attainment. And I'm going to zoom in on this values by sample number. So what you're seeing here, just to walk you through quickly, you're seeing the AUC over 24 hours with highlighted between 400 and 600 by level that's drawn. So what's the AUC that we're getting with the first level, right? Which would tell us how our initial dosing is doing. What is it with the second level, which tells us how we're doing with our adjustments to the first level, and so on, right? Third level, fourth level, fifth level. And what you would hope to see is that, you may not be perfect when you start out, but as you progress with successive levels, you should get closer and closer to having the center point of your box and whisker plot here be the center of your target range. And you should also hope that it gets tighter and tighter as you go, right? And you see that over the first 4 samples. You can see here actually with the 5th and 6th sample of this organization, it starts to get a little potentially spread out more. And what we found is this is actually pretty common, and it tends to be because patients who require more samples, 5, 6, 7, 10 samples, are the ones that are having trouble. They had the AKI event, or they required us to get multiple levels to see where they're at, and things are changing. And so things can get a little wonky as you get out there. But you can see from this organization, over these first 4 samples, we start in these box whisker plots that they start a little bit on the low end, but the median is in range, and we get tighter and tighter and closer to the middle as we go. Looking now at the red box that we have on here, this is our true baseline things for what we want to investigate in this case. And so we want to know how we're doing with initial dosing on our patients. And you can see here, as a baseline for our general population at this institution, things that are in range with initial dosing, around 50%, and around 40% are low. So that raises the question, maybe this is just confirmation bias. Maybe there isn't an issue with this population. It also raises other questions, right? Why are we low to start? And there could be a whole bunch of reasons. It could be an issue with the model, but it could also be the fact that maybe we're targeting too low. If you have people that we say 400 to 600, but you're targeting 420 every time, you're going to expect that you're going to more often than not have some people who are on the low end just because of that initial spread. Or this could be a case to examine how often we're doing loading doses. Maybe if we did loading doses more often, we would get people up to steady state faster, and we would be able to get them more in the therapeutic range. But regardless, for our purposes in this story, what we're seeing here is we have baseline data saying 50% are in range, 40% are low. Now, what we talked about earlier with this real-time analytics, which again happens instantaneously, you open this and click it and it's right there for you, is we're also able to do that drill down. And I'm going to zoom in on the applying filters over here. And here you can see a whole series of ways that you can filter your data: age, weight, serum creatinine, BMI, whether they had an acute kidney injury or not. And then finally, treatment tags, which is this final just wide open area of any customization you want. The treatment tags is something that you can apply to patients in real time as you're working on them. And we have some basic ones in there, like where they are, the indication, but these are customizable to whatever you want. You can, if you want to examine different ethnicities, you can do that. If you want to examine the floor, again, we talked about whether they're co-administered piperacillin and tazobactam or cefepime. You can look at that. Anything you can think of that you want to examine, you can have these tags in real time and then filter by them. So in our case, what we care about is this population who is elderly, we'll say greater than 65, and low serum creatinine. We're going to say less than 1. So I'm going to filter by that, right? Age greater than 65, serum creatinine less than 1. And we'll see what we get. And I'll flip right over to a zoomed in version of that same chart. So take a look here, right? In range, 40%. Low, 50%. And now we only have 87 patients, right? So this may not be statistically significant, but it sort of confirms our suspicion, and we have a signal now, right? We detected a signal. Something may be going on here. I'll go back to our historical, right? Historically, this is the endpoint. You get your MUE, your DUE, and you're like, yeah, looks like there might be a problem here. We should think about doing something about it. Maybe just fudging our data, not fudging our data, fudging our interpretation of things early on and just dosing higher than we think is going to be needed for this population, or doing some more examinations along the way. But in this case, with this precision dosing intelligence cycle of continual improvement, this is where, again, it gets to the cool part that that made me ditch my clinical job that I loved for 10 years to be able to join this process. So we're going to move to the third pillar now. The third pillar is continuous learning. And there's no product associated with this. It's a process. And the process involves our data science team. And so what was done in this case was we heard this message from multiple different sites. And they said, we're finding that in this population, in these elderly patients with low serum creatinine, we have to adjust. We're undershooting. consistently. And so we investigated a little bit, and we talked to multiple organizations around the country, around the world. And some said, yes, we had it. Some said, no, we don't have that problem. And we tried to figure out a common thread. And it turned out the common thread was organizations that were using this GOTE model as their basis were more likely to have this issue. And so we said, well, let's look at the model and let's see if there's something going on there. And our data science team started to look into it. Here's a summary of the model. This is actually available in the InsightRX Nova decision support platform. There's a lot of information on here, but I want to point your attention to two specific areas. One, this top area describing the model. And what I want to point to here is the fact that this is a good and robust model. 1,800 patients at 2 sites with nearly 3,000 samples. That is a large data set with a good, robust sampling in it. You can see that for the age group, the median was 57 with a range of 17 to 101. That probably reflects pretty well the general population you'd see. Serum creatinine with a median of 1.3, range of 0.2 to 16.6. Again, in terms of being impacting the general population and reflecting the general population, this is a good model, and it's one of the reasons why it's one of the top models that we have available right now. What we don't have here is any breakdown of how it performs in this subpopulation that we want to look at. And that's common practice with models. You bring a model out and you can only do so much with the data that you have at the time, and then you have to publish it and people use it for what it is. And there's a hope that maybe we can improve as we go on. But a lot of times it's just static. The other thing I want to point to you over here is this bottom section. And this is sort of written in kind of a jumbly form, but the summary of what we're seeing here is that for this model, they rounded up serum creatinine to 1 in patients who were age 65 or older, which honestly is sort of validating for us because a lot of us do something similar to this. And it's good to see that a model found that this practice is supported and incorporated into their model to say that this is something that will work well. So we had more questions then, right? So we wanted to see, if they're doing this rounding up, how did they find that this was the right answer, and why does it potentially not seem to be doing so great in our practice? Unfortunately, as I said, they didn't have a subgroup analysis in the original model, but we were hoping to see the investigation that we could on here. I'm sorry, I want to point out on here, and I'm not sure if you can see the highlighting on this, but we went to investigate the original paper to find out the validation of how they came to this decision. And you can see that serum creatinine values were truncated to 1 if the serum creatinine value was less than 1 and the patient's age was greater than 65 for creatinine clearance. And then they say that the correction in the elderly mirrors clinical practice and is applied to correct for falsely depressed serum creatinine in elderly because of muscle mass or decreased protein intake. So what we found in reading the original paper was that this model, which again is a fantastic model, I don't mean to belittle this model at all. It is one of the top models available. They used this rounding factor to mirror clinical practice. They did it because it's what's commonly done, and they found that in the general population, it seemed okay. But there was no regression analysis to determine whether this is the right decision or not in the paper, which again is common in models. You can only do so much with the population that you have. So what we want to do potentially is see if anything can be done in this subpopulation. Now, this is sort of a summary, right? On the left side, the population we care about is patients who are age of greater than 65, non-obese patients because things can get messy when you're in that area, and a serum creatinine of less than 1. In the original paper, the total number of patients were 1,812, which again is a good, robust model. The number of patients meeting this criteria is hard to say. I think we would be generous to say that 25 or 20% of these patients met criteria, so 400 even would probably be generous to say that. And we also don't know how the model performed in this patient population. It could perform very well in the general population, but not so well in this subpopulation. The good news is, with the continuous learning process, at InsightRX, at the time when we decided to investigate that, we had more than 50,000 patients available in our database de-identified from hospitals and organizations around the country and around the world. And patients specifically meeting these criteria of age greater than 65, BMI less than or equal to 40, serum creatinine less than 1, we had 6,206 patients meeting those criteria. So we have the potential now, with ongoing data and improving data, to validate the model and see if there's room for improvement. And that's what our data science team did. They looked at two versions of this model. So on the left is the original GOTE model. GOTE OG is what we called it, which the model as it came packaged from the publication. And on the right, what we compared that to was what we called GOTE NR, for no serum creatinine rounding. And the only change was to remove this rounding factor of serum creatinine. Performed a data validation analysis, and here's what they found. For those who are familiar with these type of things, we have MPE, MAPE, and RMSE. For those who are not familiar with statistics, I'll walk you through it. We have light green is the original GOTE model. Teal is the GOTE non-rounded model. And you can see on the left side, we have bias. We want bias to be 0. And taking a look, you can see that whether you're before we have levels, initial dosing, or with levels afterwards, removing that rounding factor moves the bias closer to 0. It improves it. Likewise, on the right side, we have these two measures of imprecision. We want imprecision to be 0 because we want precise. No matter how you cut it, removing that serum creatinine factor improves the bias, improves the precision in this population. So, holy cow, right? We have a moment here where we're saying, first, maybe our serum creatinine rounding that we've been doing all along might not be the appropriate thing to do. But at the same time, we have this model, which is a good model that, not an imperfection, but there's a way possibly to improve this model. And people could be doing better as they dose. So what do we do from there? Well, we are a team of scientists. So we did submit our findings to Therapeutic Drug Monitoring. And just last month, they published these findings in a letter to the editor. But this was, we originally had these findings months and months ago. If you've ever tried to submit a manuscript, you know that between submitting and revisions and the time between acceptance of publication, we're talking months of time, and we're just sitting on this data in the meantime. So what we also did was we coded up the model within InsightRX, called it GOTE Unrounded, did some extensive and rigorous validation testing to make sure it did what we thought it was going to do, and then put it in the platform, offered it to people who are using the platform along with everything else for free, and sent a message to them saying, hey, we have an improvement, and we encourage you to use this improvement if at all possible. So that the next time someone goes in, here's Emelsa kicking. This is Stella's twin sister. We go in and we click on Show Regimen Options. And look, we have a new model. Adult General Population Modified GOTE. This actually says InsightRX data on file because at the time I created this, we didn't actually have the publication yet. So we load that up. We have the model now loaded in. And when I look at the dose assist recommendations now, take a look. 750 Q12 is recommended for an AUC of 450. It's done, right? We've improved the model in real time so that now the data available is now enhanced. Our new best data available has changed with these findings. So you can see now this cycle, right? We start with dose and decision support, using clinical practice with the best data available. The findings from that lead into real-time analytics where we can view things on these patients and explore what's going on to see if there's opportunities for improvement and find a signal. And that signal can lead into continuous learning, the third pillar, where the signal can be expanded to a massive, massive data set, see if it's real or not, and then potentially improve the underlying models. And that feeds back into dose and decision support, where now these new models become the new best data available. And this example was vancomycin, but you can imagine this can apply in all different types of things, right? I mean, drug companies, when they develop stuff, only have so much data and they put it out there. And we can find out more and more as we go with patients to be able to improve things. Drugs that are out there right now, aminoglycosides or azole antifungals, anything where we do monitoring. Honestly, GCSF and the impact on neutropenia, anytime you have a dose-dependent response, this system can be used, this precision dosing intelligence, to just have this cycle of continuous learning that continually improves our patients. And I'll note lastly, this cycle from noticing this to the turnaround and getting in the platform again was weeks. This is why I dropped my clinical position and came, because of the promise of this, to be able to improve things in the course of weeks, which I think is just fascinating. Obviously, I'm drinking the Kool-Aid, but I hope you all can see the promise and the excitement that this has. That's the end of the formal things I have on here. I guess we'll open it up for questions, and I'll move to Andrew for that.
Hi, John. Thanks for the great talk. We have gotten a question or two in the questions and answers tab, and I'll ask everybody to, if they have questions they want to be answered now, please type them into the Q&A tab, and/or you can stop by our virtual booth and schedule or see a live demo or schedule one. We have about 5 minutes for Q&A, John. So first question here is from Elizabeth Covington. She's asking, is there a capability to round the serum creatinine for initial or empiric dosing if desired? And I know you touched upon this quite a bit, but
Yeah, I saw this was asked early on as well. It's a good question. So I think in the platform, we don't have the ability to round it up. We do have the ability in other ways to set an EGFR cap. So you can set to say that, I don't think anyone's going to have a creatinine clearance higher than 150. And that's a very common thing. That's actually also included in the GOTI model and proved to perform well to have that cap on there. I think we talked about during the talk, I think since then, the fact that the GOTI model in and of itself, the original one, does round serum creatinine up. So that capability is there if you choose to make it available. Through the talk, I think you found that it turns out that may not be the best practice, but there are capabilities to do that if you so desire, to customize that in. It's a great question.
right. And to be clear, the original GOTI model is still also available in addition to our modified model?
That's correct. Both versions of the model are still available on the platform.
Okay. Let's see. Don't have a lot of other Q&A now. Is there anything else you want to touch upon or
No,
go into more detail?
A great point. Obviously, I blabbed on longer than I want to because I'm so excited about these things. But I think it's just important to emphasize that cycle and the fact that I think we're used to, especially myself as a pharmacist, we're used to working with what we have available, and we're used to working around things and working with it. And it's something that I love about pharmacists. But I think this idea of precision dosing intelligence sort of opens the door to say, yes, we work with what we have, but we can also improve upon it. And we can open our minds to the ideas that we don't have to deal with something that's static. And if there are ideas for ways to improve in this world, and InsightRX is one example of it, but there are lots of opportunities going on where we have the opportunity and the technology to be able to improve things in a very short amount of time. And so I encourage everybody to, if you have ideas, share your ideas. I think a recurring theme in a lot of conferences is publish, get things out there, share ideas. The world is getting small. We can work together very easily on these things. And this is just one example of that.
John, actually, another example of feedback we've gotten from our customers and implemented is the dose assist feature that you were demonstrating or showing very early on. We have a couple minutes left if you want to describe how we got that feedback originally and what went into building this.
Yeah, that's a great point. So dose assist, I sort of touched on it, right? And I said that we got feedback that people were saying, we wish that you gave us the right answer. So the process that we used for it was we reviewed basically every possible iteration of regimens, 250, 500, 750, 1,000, 1250, on and on and on, up to 2,000, and every possible interval. And we reviewed data available to see what type of things are being used in practice. And we also sort of used our own clinical considerations to say, what is logistically feasible, what is in a pre-made vial gives it a bonus, and what is going to be the things that people like to use the most. And we used that to create what we call the happiness score, basically, which is ranking all these things in a product evaluation, and then sent it out to our customers sort of in a beta phase and said, does this work? And there are opportunities to tweak a little bit, but we found that in the end, we got this system where, again, we're not going to say that the one that's on the first line is the one you want to use all the time, but chances are among the things that we provide, one of those options is going to suit what you need. And if not, there's always a custom field to enter whatever you want. But the goal of this was to decrease the amount of time someone had to manually enter and decide on their own which regimen to use and just provide the best ones for them.
Great. We have one more question that just came in from Geeta Shah. If you could say a word about good trough level, I think that's in the context of AUC targeting.
Yeah, and this This has some controversy and can be a whole talk on its own. So, traditionally, we have said that 10 to 20 was needed. In the 2009 guidelines, it was thought that 10 to 20 was needed to avoid MIC creep for troughs of less than 10. And speaking specifically about MRSA and Staph aureus, obviously for strep, there's whole other things, even endocarditis, we can get troughs sometimes even as low as 5. In terms of recent data, there's been a lot of new questions about this and whether having troughs below 10 truly does lead to an MIC creep, or whether there's just other factors going on in terms of exposure. People can speak from different clinical practice. I can sort of speak, I suppose, off the record in my clinical practice while I was practicing. In pediatrics, we found that oftentimes, actually there's a great study, combined of studies out of San Diego and Seattle, saying that 8 to 12 is a reasonable way to target in pediatrics. And so far, I have not read of anything about MIC creep in those populations. But it is sort of up in the air.
Okay, great. Thanks. John, we're just about out of time, and thank you to the audience for attending, and a reminder that you can stop by our booth if you want to continue the chat or see a live demo or schedule a demo for the future. Thank you very much, and have a great day.
Everybody. Have a good one.